Hi guys. So in this lecture we are continuing on looking at plant anatomy, and now we're moving up that step in complexity. So we started with plant cells, now we're going to be talking about plant tissues. So let's quickly review plant cells and then we're going to get into this. Alright, so in last lecture we looked at plant cells. And we said the cell is the smallest unit of life. And we made a distinction between eukaryotes or eukaryotic cells and prokaryotes or prokaryotic cells. Our eukaryotes are our plant and animal cells. And importantly, they have a nucleus and other membrane-bound organelles, whereas our prokaryotes, which are our bacteria, they're more primitive, blue-green algae, they don't have a nucleus or membrane-bound organelles. So we are focusing on our eukaryotes, and we said that there are some differences between plant cells and animal cells. And those important differences are that our plant cells have a cell wall, which gives them a more rigid structure. They also have chloroplasts, which are organelles that are responsible for photosynthesis, and that's how plants are able to create their own food. And plant cells also have a large central vacuole that, while our animal cells have vacuoles, it's typically not this large central one that you see in our plant cells. So those were the differences, and then we actually got into plant cell structure. We looked at a couple different organelles. And the ones that I want to highlight, we're going to talk about right now. So we looked at the cell wall down here, right? And we said the cell wall helps protect the cell, and it provides some structure to the plant. We then moved inward, and we said inward of the cell wall was the cell membrane right here, and this is a lipid bilayer that's selectively permeable. It's like the gatekeeper of the cell. It allows some materials in and prevents other materials from coming in, and vice versa, also going out. We then looked at, let me see if we have it here. Oh, it's not on here, so I'm going to draw it in. We then had what I refer to as a tunnel, which are the plasmodesmata. And these are little narrow strips of cytoplasm that connect adjacent cells. And through these strips, we have, so it's like a little tunnel. And through these strips, these cells are able to transport materials back and forth to each other. And these little gaps in the cell wall are called pits. I just mentioned cytoplasm. Cytoplasm is the thick fluid in which all of our organelles sit. It's made up of water and some proteins, lipids. And then I mentioned that the cell membrane and everything interior to it is referred to as the protoplast. And then I started talking about a series of organelles that are all involved in gene expression and protein synthesis. And I said those organelles were our nucleus, nucleolus in a sense, ribosomes, endoplasmic reticulum, and Golgi apparatus. What is happening with these cells or with these organelles is that within the nucleus, which is the command center of the cell, it contains the DNA, all your genetic material. And that DNA is codes for your traits, ultimately. So a single strand of DNA, or a DNA is actually double-stranded, like so. And it consists of these two sugar phosphate backbones with these nucleic acid bases in between them that connect the strands. And it is the sequence of these bases. Oh man, I got some bad handwriting. It's the sequence of these bases. They are adenine, thymine, guanine, and cytosine. It's the sequence of these bases that is going to code for a particular protein. and it is the interaction of proteins with the environment that creates a trait, whatever trait you're talking about. So within the nucleus, we have this process called transcription, where we take a sequence of DNA and it gets transcribed to another molecule called a messenger RNA. That messenger RNA then travels to a ribosome, and within that ribosome, it goes through a process of translation where it gets fed through the ribosome, and a different molecule called transfer RNA binds to that sequence. And as it does that, it leaves behind a series of amino acids that join together to form a protein. That protein then gets folded up by the endoplasmic reticulum. Where am I here? By the endoplasmic reticulum, and it gets sent to the Golgi apparatus. And within the Golgi apparatus, which is like the post office of the cell, it can further modify that protein, it will repackage it, and then it will send it off to where it needs to go, either within the cell or outside of the cell. So all of those things are involved in this gene expression and protein synthesis. I didn't mention the nucleolus, but this is responsible for making ribosomes, so that's why it's included. So those organelles are part of gene expression. I then talked about plastids, so those include our chloroplasts right here, as well as these amyoplasts. The big one, the most important one that we're going to talk about is this chloroplast. They are the organelles where photosynthesis takes place. So that within the chloroplast is where the plant is able to take the energy from the sun and use that energy to combine carbon dioxide with water to make a sugar molecule. And that is the foundation of what's going to power all of our cellular processes, or all the plant cellular processes, and it's the foundation of food. So that happens in the chloroplast, and kind of as an interesting little aside, we said that these chloroplasts have an extracellular origin, right? Way back 1 billion years ago, a single-celled eukaryotic organism probably ate some sort of cyanobacteria or engulfed it. Instead of breaking it down, it just incorporated it into its body, and that cyanobacteria eventually became a chloroplast. And then we said another important organelle is the mitochondria, or mitochondrion, if we're talking about one. And the mitochondria is where cellular respiration takes place. And that's where we take that sugar molecule that's produced in photosynthesis, and we break it down to create ATP, which is then going to power all of these cellular processes that take place. And then I believe last we looked at the vacuole, and we said the vacuole is important as both a storage structure and as a way that the plant maintains turgor pressure. And turgor pressure is the pressure exerted within the cell to push the cell membrane against the cell wall. And if we don't have enough turgor pressure, the cell membrane will begin to pull away from the cell wall. So it will begin to kind of pull away like this. Yep. And as it does this, it is manifested in the plant as the plant wilting, right? It starts to lose that structural support. So again, so that vacuole is important for preventing that wilt and keeping the plant upright and rigid. And there is a membrane around this vacuole called the tonoplast that lets water in and out, and other materials. And that should be it. All right. So tissues are groups of cells with similar structure that perform a collective function. And if you kind of see from that image there, we have a nice example of how we're going to be working these next couple of lectures, right? We started with cells, that's that basic unit of life. And now we're going to be looking at groups of cells, which we call tissues. And then later on, we'll be looking at organs, which are groups of tissues that are forming collective function, organ systems, and then eventually the entire organism. Right, so we're moving up this ladder of complexity. where we started with our cells. Now we're looking at groups of cells that are all performing a collective function. Now when we're talking about plant tissues, we have two main types. We have meristematic tissues, or meristems, and we have permanent tissues. Now, meristematic tissues are made up of actively dividing cells that are going to develop into other types of tissues, and that's the big differentiation. Meristematic tissues are made up of actively dividing cells. Permanent tissues are no longer dividing, right? They are made from meristematic cells, but permanent tissues are made up of cells that are no longer dividing. They are fully differentiated. And when I say differentiated, I just mean when those cells are originally formed, they eventually, as they mature, they take on particular roles. So they differentiate into whatever tissues and whatever roles they're going to have. So our meristematic tissues are made up of cells that are still actively dividing. The meristems are actively creating more tissues. So, as a result, these meristems are found in these actively growing regions of the plant, particularly the stem and the root. Now, meristematic cells, some characteristics: they typically have thin cell walls, they have a lot of cytoplasm and protoplasm, a prominent nucleus. Whereas, like I said, with our permanent tissues, they are differentiated, they are no longer going to divide. With exception. There is a little bit of an exception here, but we're not going to get into it now. So we're going to start by looking at our meristematic tissue types. And there's three main types, and kind of this fourth one I include as well. But again, these are actively dividing tissues. These meristems are creating new cells. And the ones that we're going to focus on are apical meristems, which are found at the tips of our roots and our shoots, intercalary meristems, which are found at the base of nodes between permanent tissues, and lateral meristems, which are found really like a cylinder around the sides of our roots and our shoots. So we're going to look at each one of these, and I'll also talk about subapical meristems. They're kind of a special type that's not found in all plants, I believe. Or if they are found in all plants, they're more common and important in some particular types that we'll look at. It's actually a good question. Okay, so apical meristems. They are present at the tips of our shoots and our roots, and they are responsible for primary growth. So primary growth is the lengthening of the plant. And we can contrast this with secondary growth, which is the thickening of a plant. Right? That's going to be the responsibility of our lateral meristems when we get to it. But our apical meristems are responsible for the primary growth, the lengthening of our plant. Right? And as a result, because they're found in the tips of our shoots, they produce new buds and leaves and stem tissue, and as well as new root tissue down here. So right now you've guys got your pocket seeds germinating, or you likely have them germinating, and as you're seeing them growing, at the tips of that root and at the tip of your shoot, that stem, are apical meristematic tissue, and that's producing all the new structures initially that you're seeing growing. So here, again, you can see here's our apical meristem this is our shoot, and this would be our apical meristem at the tip of our root. With our roots, there's also a root cap, which we'll talk about next lecture, that just provides some protection of this meristematic region. So as I said, our apical meristems, they increase the length of the plant, right? That's our primary growth. Some apical meristems eventually develop into a terminal inflorescence, which would be a flower grouping when we talk about inflorescence, or a terminal flower. Others remain vegetative. Another kind of interesting aspect of our apical meristems is that they display dominance, apical dominance. So when you have an active apical bud growing in your shoot, As this stem is growing, it's also producing other buds, and those buds are dormant tissue that can develop into other shoots or leaves. But as this apical bud is present, it produces a hormone, which we'll talk about, that prevents the active growth of these buds. It keeps them dormant. However, when that bud is removed, that apical bud, you will start to see these lateral buds start to develop and grow. So this is something that can be used by propagators if you are trying to create a bushier shrub as opposed to one that's more tree-like. One of the ways you can do it is to remove that apical bud, and that will promote the growth of these lateral buds. So something to be aware of. When we talk about hormones, we'll get a little more into this, or we'll highlight this again. But again, it's an aspect of these apical meristems. Okay, now we also have subapical meristems in some of our plants. And this is, they are a region of cells found a few micrometers, so very, very close to where the apical meristem is found, right? Just behind it, if we're thinking about a stem. Now, our subapical meristems are common in rosette-forming plants. So think of things like beets or carrots, turnips, evening primroses, which is a wild plant that you'll see a lot. Now, these types of plants, they can be often biennial. So they grow one year, don't produce flowers. The second year, they're going to produce flowers. They typically lack tall stems when they are first producing leaves, right? So they're going to form this basal rosette that you'll see here and here. And eventually, later, when the time is right, and they're going to initiate flowering, they're going to develop this stem that's going to grow out of here. That's going to be a flowering at the end of the stem. There's going to be flowers at the end of the stem. And that is going to develop pretty rapidly. Well, during that, one of the important regions that is helping that stem develop so quickly is this subapical meristem. Right, so you have your meristem that's producing new tissues, and then your subapical meristem right behind it is producing new cells and tissues to help speed along the growth of that stem. So that's subapical meristems. Again, this isn't as important as the main ones, but it's something just to be aware of. So we're going to move on and let's talk about intercalary meristems, right? So our meristematic tissue, active tissue, it is made up of active cells that are producing new cells. We talked about our apical meristems found at the tips of our shoots and our roots, subapical meristems found in our rosette-forming plants, basal rosette-forming plants, right below that apical meristem. And now we're going to look at intercalary meristems. And these are found in our monocots, particularly our grasses. And these intercalary meristems are found between sections of permanent tissue, and they're found at the base of nodes. So nodes are just attachment points for a leaf or a bud or a branch. Right, so right here, this would be a node right here where this leaf is attaching to the stem. So, and this is where you will find your intercalary meristems. Right, example is right here. Right, that is intercalary meristematic tissue. Now, the importance of our intercalary meristems is that they allow for damaged leaves or damaged tissue to rapidly regrow. And why this is important in our grasses, if you recall, our grasses have evolved to be grazed upon, right? Number of animals graze upon grasses. So these intercalary meristems allow these grasses that get grazed upon frequently to quickly regrow leaf or other stem tissue that could be damaged, right? If they just depended on their apical meristem, if a cow comes along, eats the top of the plant, it's going to eat that meristematic tissue, the top, right? And the plant's going to not be able to regrow. But if you have a series of meristematic regions throughout the stem and by the leaves, then when it gets grazed upon, there are these regions that can begin producing new tissues. So, very, very important in our monocots, particularly our grasses that get grazed upon. And lastly, when we're looking at meristematic tissue, we have our lateral meristems. Now, our lateral meristems are only found in dicots and gymnosperms. And they form essentially a cylinder around your roots and your stems. And they're responsible for secondary growth, which is the thickening of a stem or a root. Remember, primary growth, lengthening, secondary growth, thickening. And we have two types of lateral meristems. We have vascular cambium, which is tissue that's going to give rise to secondary xylem and secondary phloem, and we're going to go into what xylem and phloem do. They're very important. And we also have cork cambium, which produces bark. So as an example, here is a stem of some dicot. And if you look, we do a little cross-section of our stem, we see that we have primary xylem, primary phloem, and between it we have vascular cambium. So this primary xylem and primary phloem is produced by the apical meristem, and we'll talk about that in our next lecture. And it also produces vascular cambium. And like I said, vascular cambium is responsible for the thickening of our stems, right? So as this plant is going to grow, where's my thing? As the plant is grow, this vascular cambium right here produces secondary xylem, so new xylem, and secondary phloem, new phloem on either side of it. And in that process, it increases the thickness of that stem, right? And you can see it getting thicker and thicker each year, right, as it produces more secondary xylem and secondary phloem. Think of like tree rings, right? That cross section where you see, I'm sure you've all seen a cut trunk of a tree, and you see the rings of growth. That is what you're seeing here. It's that particularly when you're seeing those rings of growth, you're looking at the rings of this secondary xylem that gets produced. So that is a vascular cambium, which is a type of lateral meristem. We only see this in our dicots and our gymnosperms. You don't see this in our monocots. And it's producing this secondary growth, this thickening of our stems and our roots. It's also, besides vascular cambium is the most important part, but there is also, which I don't think is, you don't see it here, but you will also have cork. Oh, there it is, right there. Cork cambium, another type of lateral meristem that is going to produce cork or bark tissue, right? Same idea as our vascular cambium. Okay, so that's meristematic tissue. That is the actively growing tissue that is made up of cells that are actively dividing. That's the one main category of plant tissues. The other main category is permanent tissues. So these are tissues that have been formed from the meristematic tissues, but they're no longer actively dividing. And within our permanent tissues, we have two types. We have our simple tissues, which are made up of only one cell type. And our examples are parenchyma, sclerenchyma, and collenchyma. Kind of crazy names. And we have our complex tissues, complex permanent tissues, which are composed of multiple cell types. And the examples are xylem and phloem, that vascular tissue that's produced, at least produced from the apical meristem and then produced by the vascular cambium. So let's take a look at them now. So we're looking at our simple permanent tissues. So again, these are tissues that have been produced from meristematic tissues, but they're no longer actively dividing. They have been differentiated into whatever tissue type they're going to be. And within that permanent tissue, we have our simple tissues, which are just made up of one cell type. And within our simple permanent tissues, we have parenchyma tissue. Now, parenchyma tissue is kind of like, it's kind of like an all-around, does a whole bunch of different jobs type tissue. I can't think of the proper word that what I'm trying to think of here. But it essentially has multiple jobs, plays multiple roles within the plant. Right? It's used in photosynthesis. This is tissue that photosynthesizes, is found in the leaves and in some stems. It's found throughout the complex tissues. It can be used as storage tissue, as I have up there. It's a principal tissue in the cortex, which is this region between the epidermis, which is the outer layer of your stem or your root, and the phloem. And there it acts primarily as a storage type tissue. So it can take on a number of roles in the plant. It's kind of this all around, does a whole bunch of stuff. It is typically made up of thin-walled cells that have those large vacuoles, those large central vacuoles. There's some intercellular space present between the cells, right? So that just means there's some gaps between these cells. And like I said, it can be found, it's this all-around tissue. It's found in all plant parts, photosynthesizes, acts as storage tissue, takes on a lot of roles. Now, another simple permanent tissue is this sclerenchyma tissue. Now, this is much more specialized for providing structure, right? So our sclerenchyma tissue is composed of these thick-walled cells, and here in these cells, those protoplasts eventually die. So all in this tissue, what you're left with eventually is just a bunch of cell walls that are very, very thick cell walls that contain a molecule called lignin, which is going to provide more hardness to this tissue. So our sclerenchyma tissue is going to be thick cell walls that are hardened with lignin. There's no intercellular space between them, and they are providing tough structure, right? We find sclerenchyma tissue in stem and bark tissue where you need a lot of structure. Another example that I got right up there, in walnut shells. Think of that walnut shell, how tough that is. That is because it is composed of this sclerenchyma tissue. It's this hard, tough tissue that is made up of mostly dead cells that is going to provide structural support. And then we have collenchyma tissue. And again, this is also a type of tissue that's going to provide structural support. However, it's a little bit more flexible. It's not as hard and rigid as that sclerenchyma. Right, so you'll find this in regions of the plant that need support, but then also need a little bit of flexibility. So you'll find it in stem tissue. You'll find it in petioles, which are the stem of the leaf. And think about how that's a little flexible. Our cell walls here are thickened, just like our sclerenchyma tissue, but they're thickened with cellulose instead of that lignin, so they're not as rigid. And like I said, they provide support, but also a little bit of flexibility. And again, they're kind of this intermediary between our parenchyma and our collenchyma in terms of intercellular space, right? So if we look at our collenchyma, we got these thick cell walls, no real intercellular space. Parenchyma, we got a lot of intercellular space. Our collenchyma are like an in-between, right? So to summarize our simple permanent tissues, we have our parenchyma, which is like our all-around tissue, provides storage, it's used in photosynthesis, you'll find it in all parts of the plant. We have our sclerenchyma tissue, which is specialized really for structural support. It's just cell walls. Eventually when it matures, all the actual cell parts die. Protoplast dies. And then we have our collenchyma, which is also used for support, but it's not as rigid, it's a little more flexible. There we go. Those are our simple permanent tissues. Other types of simple tissues, just to be aware of, the epidermis, I mentioned it before, that's a single exterior layer of cells that are going to protect the stem, leaves, flowers, roots. It's the outer layer of cells of our plants. Think of epidermis, you have an epidermis of your skin, it's the outer layer. Same, the epidermis is the outer layer of cells for our plants. It's often covered in a waxy material called cutin, except in the root epidermis, and that keeps everything waterproof. Other simple tissues is cork tissue, which is the outer layer of bark. And here our cell walls are waterproofed with our bark layer with another waxy material to keep it waterproof called suberin. And just like with our sclerenchyma tissues, our cork tissues, they lose their protoplasts and die, but their cell walls remain. Okay, so now lastly we're going to look at complex tissues. And again, these are permanent tissues, But what separates them from our simple permanent tissues is that our complex tissues are made up of multiple cell types. And we have two very important tissues. We have our xylem tissue and our phloem. Both of these tissues are part of the vascular system. And when I'm saying vascular, this is the system that's going to transport, in this case, water, sugar, minerals throughout the plant. So our xylem tissue conducts water and minerals from the roots to all other parts of the plant. So it goes from the roots upward to the plant. And our xylem is composed of 4 different cell types: vessels, tracheids, fibers, and those parenchyma cells. Our phloem tissue, another thing about the xylem tissue, it's a one-way street. So water and minerals come from the roots up to the plant. It doesn't transport it back down. Phloem tissue, two-way street. Can move up and down. And our phloem tissue conducts sugar, or photosynthate or glucose, whatever you want to call it, and other metabolites from the leaves, flowers, roots, and storage organs throughout the plant. And our phloem is composed of sieve elements, which consist of sieve tube members and sieve cells, companion cells, fibers, and again, those parenchyma cells. Now, the way I always remember it is food phloem. So the phloem tissue is transporting essentially the food of the plant, the glucose that is produced by the plant, and the xylem transports the water. So if we look at our xylem is made up of those 4 tissues. So those xylem vessels, tracheids, fibers, and parenchyma. The vessels are long tubes that are made up of these vessel members and are joined end to end, and they essentially form like a big straw, right? So here are our xylem vessels, and these cells all connect, and the cell walls at the top and bottom of each vessel member dissolve, and this creates a long tube. The vessel cells are dead at maturity, right? And xylem vessels are only found in our angiosperms. You won't find these in our gymnosperms, right? So these xylem vessels create this long tube that's going to help transport water. There are also present tracheids. Tracheids act very similarly to our xylem vessels. The main difference being, is that these cell walls don't divide. So the water has to travel through past these cell walls instead of there being this nice open tube. Tracheids are found in our angiosperms along with the xylem vessels, but within our gymnosperms, we only find tracheids. We don't find these xylem vessels. We also have, as part of this tissue, xylem fibers, and those fibers are providing structural support. And what types of tissue, simple tissue, provide structural support? Our sclerenchyma tissue, right here. So these fibers are providing structural support, keeping these xylem tissue supported. Good one, Zach. So it keeps that supported. And it's made of that sclerenchyma. And then also mixed in here, we have parenchyma cells that are going to act as kind of storage tissues around our xylem tissue. And lastly, we have little pits in our vessels and our tracheids that is going to allow the movement of water between cells. Remember, those pits are those gaps in those cell walls, right? Often in our living cells, it's where you will find plasmodesmata, those little narrow bands of cytoplasm that connect cell to cell. For here, where these cells are dead, they don't have cytoplasm anymore, but they still have these pits that allows water to move from cell to cell. All right, and then you have your phloem. So again, food phloem. Phloem is the tissue that's going to be transporting sugars, primarily. Right, so transporting sugars throughout the plant, and those sugars get broken down through cellular respiration to power all these cellular activities of the plant. Now, our phloem is composed of sieve elements, which, depending if you are an angiosperm or gymnosperm consist of sieve tube members or sieve cells. An interesting thing about our sieve elements is that they lack a nucleus. Remember, the brain of the cell, command center. And they have very few organelles. So they rely on these companion cells that you see here for most of their metabolic needs. So these companion cells actually produce the energy and the other types of metabolic processes that are going to allow these sieve elements to do their job and to continue to live. So like I said, if you are an angiosperm, your phloem is composed of sieve tube members, whereas if you are a gymnosperm, your phloem is composed of sieve cells. They both are very similar. The main distinction is that your sieve tube members have a sieve plate at the end of them, whereas you don't have that in your sieve cells. And again, with our phloem, this is transporting your sugars, your other organic nutrients. It goes both ways, so it can be transported up to the leaves or down to the roots. Besides those sieve elements, we also have fibers, just like our xylem, and those fibers are made of those sclerenchyma cells, right, to provide structure. And then you also have parenchyma cells as part of the phloem structure that, again, are going to provide storage. Okay. I think that is enough. So that is an overview of plant tissues. Again, it may seem like a lot because it's a lot of new, probably terms that you haven't heard before, but it's really not. I mean, it's a simple differentiation between your meristematic tissue, which is your growing tissue, actively dividing tissue, and your permanent tissue, which is your no longer actively dividing tissue. And then within those, there's some other breakdowns. But it's not too complicated. In our next lecture, we're going to look at the development of stems and roots. And as part of that, we will be talking about all these tissues again. So you'll get to see this again in the next lecture. So that's it for now. Hope you guys are doing well, and I'll see you at the next lecture.